everyone, welcome to the next meeting of Food Biotechnology class. Still with me, Pujiati, the lecturer of Biology Education Department, Universitas PGRI Madiun, Indonesia. In this meeting, I will discuss about the food safety risk in traditional fermented food. So, in this part, I will talk about some topics. There are the first one, contamination of raw or fermented products with pathogenic bacteria. And the second one is toxin and toxic compounds producing microorganisms, including mycotoxin risk and biogenic amines. 3. Chemical contamination. And the 4. Antibiotic resistance of microbes from traditional fermented foods. Okay, let's move to the first topic. Southeast Asia, especially in Indonesia, is well known for traditionally fermented foods. However, these products are generally still produced at small scale following traditional procedures. Nowadays, consumers are particularly aware of the health concern regarding food additives. The health benefit of natural and traditional food processed with no added chemical preservatives are becoming more and more attractive. Therefore, their confidence towards safety and quality of Asian fermented foods is low. Major food safety concerns are related not only to food production methods, but also to how foods are processed, stored, sold, and consumed. The main factors affecting the food safety not limited only to the improper use of chemicals such as pesticides or antibiotics, but also to improper processing and handling during storage, which could provoke the accumulation of toxic compounds such as mycotoxin or biogenic amines. Recent studies from 2007 to 2019 concerning the detection of pathogen and chemical contaminant in some of the most risky products are reported. Okay, let's start with the first topic about the contamination of raw or fermented products with the pathogenic bacteria. In Southeast Asia, consumers' confidence toward fermented products is low, especially because of the microbial safety risks of the raw material such as meat and vegetable. One of the riskiest traditional fermented meat products is an uncooked pork sauce eaten after the short-lasting fermentation. To elaborate this product, pork is bought in the wet market or supermarket, and then the minced meat is mixed with herbs and spices such as garlic, guava leaf, fresh chili, etc. The finished products are poked into banana leaf to provide an anaerobic environment for the fermentation process and to inhibit entry of potential pathogenic microorganisms. The products are stored at room temperature for spontaneous fermentation for several days. The highest risk of this product is related to meat since this fermentation sauce is uncooked and unheated. In pork raw meat from Southeast Asia, Escherichia coli, Salmonella, Campylobacter, Listeria monocytogenes, and Staphylococcus aureus are the main foodborne pathogens, and they are frequently found in intestinal tract and feces of food animals. These bacteria, as well as antibiotic-resistant genes, can be horizontally transferred to human through direct and indirect contact with the source of infection, which can be animals or contaminated foods. In raw food, in the examples given in Table 1, these bacteria are found with at least 10% probability in contamination. The probability could rise up to 83% depending on the species, the raw food, and the origin of the sample. In fermented meat, the contamination is still very high in the examples presented in Table 1, and they even reached 100% in the neighboring region of India, although in one study not presenting whether fermented meat was cooked or not, contamination ranged from 0 to 19%. So this is the Table 1 about the contamination of raw and fermented products in Vietnam, Thailand, Cambodia, and Northeast India. Cross-contamination between species also have been observed as shown in Table 1, although invasion of Campylobacter concerned mostly poultry and poultry products. The species was also found in swine carcasses and retail product as the meat could be contaminated with feces at the slaughterhouse and processing facilities during the evisceration process, leading to contamination of food products. Next, we will discuss about the next topic about the toxin or toxic compounds producing microorganisms, including mycotoxin risk. Mycotoxin are toxic secondary fungus metabolites mainly produced by 5 genera of filamentous fungi, for example, Alternaria, Aspergillus, Cladosporium, Fusarium, and Penicillium. These molds can be produced different types of mycotoxins. Once they contaminate food and feed, they cannot be removed safely. This means that once food are contaminated, human exposure is almost certain if the foods go into the market. FAO reported that 25% of the world crops are affected by mold or fungal growth, with losses of around 1 billion metric tons of food products annually. Economic losses occur because of the first one is yield loss due to disease induced by toxigenic fungi. The second one, reduced crop value resulting from mycotoxin contamination. And the third one is losses in animal productivity from mycotoxin-related health problems. And the last one, human health costs. Moreover, additional costs associated with mycotoxin also include the cost of management at all levels, including prevention, sampling, mitigation, litigation, and research costs. These economic impacts affect all along the food and feed supply chains: crop producer, animal producer, grain handler, and distributors, processors, consumer, and the society as the whole due to the health care impacts and productivity losses. Nowadays, more than 400 mycotoxin metabolites have been discovered, but the 8 most important mycotoxins with worldwide relevance in regard to public health are aflatoxin, ochratoxin A, fumarisin B1, zearalenone, deoxynivalenol, nivalenol, and T2 toxin and patulin. Mycotoxin can contaminate various agricultural commodities and particularly cereals and legumes such as wheat, barley, rye, oats, rice, maize, peanuts, alfalfa, clover, beans, peas, chickpeas, lentils, lupin, mesquite, carob, soybeans, tamarinds, and some other cereal grains. They are normally used as substrates for traditional fermented food in Southeast Asia. Those substrates can be contaminated with mycotoxin either before harvest or under post-harvest conditions, resulting in mycotoxin contamination in the finished products. Mycotoxin is relatively stable to cooking and processing temperatures. Based on the figure 1, fermented foods from Southeast Asia exhibit several safety risks in a similar way to raw agricultural products. The presence of pathogens is one of the main and immediate risks as it contaminates many clean samples during slaughter and handling. However, the presence of chemical, biogenic amines, mycotoxin, and parasites can be more difficult risk to take into account as it can be more hidden with a long-term effect. Risk could be greatly diminished by the use of good agriculture or manufacturing practice, especially concerning pathogen and chemical contamination and antibiotic resistance. For mycotoxins, good producing and storage practice could also help a lot, and for biogenic amines, a survey of strain present during fermentation might be required as the first step before strategies to avoid their development. For all food safety concerns, starter can also be used for biopreservation and bioremediation purposes and thus decrease risk. The next toxin or toxic compound producing microorganism is biogenic amines. Biogenic amines or BAs are low molecular weight nitrogenous compounds naturally present in animals, plants, and microorganisms where they play several functions including gene expression, regulation, cell growth, and differentiation. BAs are divided into alphabetic, aromatic, and heterocyclic. The occurrence of some BAs in fermented foods such as fish, meat, cheese, vegetables, and wines has been widely described. Unfortunately, the consumption of food or beverages containing high amount of BAs is a risk for consumer health since they can have toxic effect. The BAs encountered in fermented foods are mainly produced by microbial decarboxylation of amino acids. The main microbial group associated with BAs accumulation are several gram-negative, including Enterobacteria and Pseudomonas, and gram-positive bacteria, including Staphylococcus, Bacillus, and lactic acid bacteria. The presence of decarboxylase positive microorganism is not the only factor influencing BAs accumulation in foods. In fact, specific environmental conditions are required. For example, availability of BAs precursors, presence of proteolytic enzymes involved in the release of the free amino acids. Other factors involved are raw material characteristics in terms of composition, pH, ion strength, physicochemical parameters including natrium chloride, pH, and ripening temperature, and processing, storage, and also distribution conditions. Several traditional fermented foods from Southeast Asia, including fish sauce and fermented soybean foods, are characterized by the high amount of BAs. The most popular fermented soybean foods are produced through bacterial fermentation. The BAs composition of these products is reported in Table 2. It should be noted that for doenjang, the very high maximal concentration detected comes from one isolated study, and most studies report values below thresholds. Okay, this is the Table 2 about the minimum and maximum BAs contained in some fermented soybean product. Next, this is the table about the biogenic eminence content of Indonesian fermented soybeans products, that is tempe. Okay, next topic is about the chemical contamination. The chemical contamination including the pesticides and heavy metals. In the past century, agriculture has increased productivity through 287 mechanization, fertilization, pesticides. Furthermore, intensification of cash crop production and conventional agriculture with fertilizer and pesticides impair local resources, including soil fertility and biodiversity. Some trace elements, for example iron, potassium, etc., have undergone historical decrease in food in Finland, United States, and United Kingdom. This decline was attributed to varietal selection based mainly on yield and soil degradation due to intensive agriculture. Great transitions are at work in Europe. The Ecopito plan is implemented with the purpose of progressively reducing the use of pesticides. With international trade, food products are imported from numerous countries where the environmental rules do not correspond to European ones. In Northern Europe, foods imported from Asia contained 111 distinct pesticides. In some cases, concentration exceeded the maximum residue levels, with leafy vegetables particularly concerned. In 2010, among 245 plant foods from Phnom Penh markets, 15% of the long beans and 95% of the kale contained noticeable levels of organophosphate and carbamate pesticide. It is true that apart from the war inheritance like Agent Orange, which still result in daily intakes of dioxin for above the WHO recommendation in Southeast Asian cultures can be potentially contaminated by chemicals and metals. Several studies have highlighted the poisoning of agricultural workers and of the environment, but the main concern for fermented food products is related to pesticides encountered in raw materials. A survey in the Red River Delta showed that pesticides used in agriculture were frequently detected in biota, leading to repeated analysis above the acceptable daily intakes in fishes and vegetables. Even banned organochlorine pesticides still persist in these environments. Among vegetables reaching the residue limits established by European Union, some vegetables used for fermentation like Chinese cabbage and some herbs are said, while some fruits, including watermelon and durians, despite the presence of chemicals, were considered as safe. The fate of pesticides in food products depend on the specific physicochemical properties of the compounds as well as the condition of preparation of foods, including presence of water, temperature, pH, etc. During fermentation, the concentration of pesticides usually decreases significantly, giving rise to degradation products. In addition to non-fermented products, fermented products are characterized by the presence of microorganisms. The presence of pesticides as well as of their degradation product tend to limit the activity of microorganisms and modify the sensorial properties of the product, but in the meantime, microorganisms can degrade these products and help decreasing the contamination of fermented products. Studies on pesticides in food are still at the preliminary level and their degradation. The risk related to their degradation product, the potential catalysis by microorganisms, thus of paramount importance, need to be further studied. Okay, the last topic is about the antibiotic resistance of microbes from traditional fermented foods from Southeast Asia. Antibiotics are either microbial secondary metabolites or the analogous compound synthesized or semi-synthesized chemically that could inhibit the growth and survival of the other bacteria. These compounds are used as therapeutic agents against infectious disease in humans, livestock, and aquaculture. However, haphazard and extensive use of antibiotics may select antibiotic-resistant bacteria. Bacteria can develop antibiotic resistance by several mechanisms via enzymatic degradation, antibiotic-targeted modification, changing the bacterial cell wall, permeability, and alternative pathway to escape the activity. Antibiotic resistance can be inherited or acquired. Inherited antibiotic resistance is exhibited by all isolated of the same species, while acquired antibiotic resistance occurs when susceptible bacteria gain the genes encoding a resistant mechanism via mutation or the transfer of genetic material from other bacteria. Generally, food contributes as an important part for transfer of antibiotic resistance in terms of antibiotic residues or resistant genes from food microflora to pathogenic bacteria. It is important to monitor the prevalence of pathogenic bacteria along with antibiotic-resistant foodborne pathogen in food chain to improve and implement food safety. For instance, for the contamination of raw product by pathogenic bacteria, as discussed above, the resistance of bacteria to antibiotic is thus also an important issue. Traditional fermented foods and beverages are popular for their nutritional balance and food security. In many Asian countries, techniques for fermenting cereals, vegetables, and meat products are well developed. Such fermentation foods are highly prized for improved nutritional and organoleptic quality as well as for beneficial microorganisms. Several studies have indicated that along with beneficial microorganisms, fermented foods can act as vehicle of antibiotic-resistant bacteria. We can see in Table 4. Therefore, through the food chain involving traditional fermented food, antibiotic-resistant genes may be transferred to other bacteria, including pathogens and commensals into the gastrointestinal tract. So this is the Table 4 about the antibiotic resistance of lactic acid bacteria involved from Asian fermented foods. We can see that in Indonesia, dadi, bekasam, and also tape ketan have observed. Okay everyone, it covers all today's materials. The study reference from Arnold et al. 2019. Thanks for the attention.